Hello, student. A very good afternoon to everyone. So, poll has been launched. Quickly answer through poll. My audio is clear and my screen is visible. So, today we are going to discuss plant kingdom. So, we are going to discuss the classification, which are artificial classification, natural classification, and phylogenetic classification. So, classification is the grouping of organisms into certain categories on the basis of their characteristics. This is known as classification. And we know that on the earth, there are millions of organisms are present, right? So, to make the study easy, we do classification. So the earliest classification is the artificial classification, is the ancient and is the primitive classification. Okay, and this classification was based on the morphological characters like habit, color, number, and shape of the leaves. The characters which you can observe by seeing, those are the morphological characters. And this classification has given more weightage or importance to the vegetative characters. Okay, and the proponents of artificial classification are Aristotle and Linnaeus. Linnaeus, he has given importance to the androecium characters. Remember this. And there's certain drawbacks regarding the artificial classification. Number one, that this classification was based on a very little information or the few characteristics. Because of that, the two species which are belonging to those having the common ancestor, they are closely related, right? So they should be grouped in the one. They should be kept in one group, but because this classification was based on a very few characteristics or the little information, these closely related species, they get separated. Second drawback is that this classification has given equal weightage to the vegetative and sexual characteristics, right? The vegetative characters could be like leaves, root, stems. These are the vegetative, and sexual characters of a plant are the flower, in which androecium and gynoecium are present. And they have given the equal weightage to the vegetative characters as well as to the sexual characters. And we know that the vegetative characters, they are more likely to get affected by the environment. For example, you have seen the plants in desert like opuntia and cactus. There the more leaves, they get modified into spines. The stem, they get modified into phylloclade, right? So the vegetative characters or the vegetative structures, they are more likely to get affected by the change in environment. But the reproductive character, they remain conserved or they are less affected by the change in environment. So, but this classification has given equal importance to the vegetative character as well as to the sexual character. So this is a drawback of the artificial classification. Next is the natural classification system. This system has given equal weightage to this system has given more importance to the reproductive character. This system was based on the natural affinity among the organism, and it has considered the external feature also as well as the internal feature. External features are the morphological characters, right? And the internal features include anatomy. Anatomy is the study of the organism at the tissue level. Embryology. Logy, always remember the logy term, it means study. Okay, so the study of embryo, that is embryology. Then phytochemistry. Phyto means plants, chemistry is for chemicals. So the chemicals which are present in plants. So this classification has taken into consideration both the external feature as well as the internal feature. This classification was given by Bentham and Hooker. Remember this. But the only drawback of this classification is that it does not include the phylogenetic relationship. Okay? Phylogeneity means the study of the evolutionary history. When we study about our ancestor, when we study the evolutionary history or the ancestry history, that is the phylogenetic relationship. For example, if the species, they are having a same or the common ancestor, they belong to the same group, right? Because they're closely related species. But this natural classification does not include the phylogenetic relationship. Next one is the phylogenetic classification. This classification was given by Engler and Prendtl. This classification is the most advanced and the most accepted classification, okay, because it includes the phylogenetic relationship among the organism. And we know that the study of the evolution, or the study of to study the evolutionary history or the ancestor history, we require fossils, right? What if the fossil evidences are not present? If the fossil evidences are not present, then we can use the numerical taxonomy to classify the organism. We can use the cytotaxonomy, we can use chemotaxonomy. Numerical taxonomy is when we assign the code to the characters. For example, there's a species, let's say PQRS. This is a species, and let's say the characters are erect stem. Let's say 5 stamens. These are the characters, okay, which I'm taking. Let's say simple leaf. These are the 3 characters which I have taken. And I will assign some code to it. If, for example, if the character is present in that species, the code is plus. If the character is absent in that species, the code is 0. So if the P species has having the erect stem, I assign plus. If it's having the 5 stamen, plus. Simple leaf, plus. If the Q species are having the erect stem, then plus. 5 stamen, plus. If they are not having a simple leaf, let's say 0. So this is how we assign the code to the observable characters. Here I have taken only 3 characters, but hundreds of characters can be taken in consideration at the same time. And then in this taxonomy, each character is given an equal importance. So now you can see, if I found a plant which is having like erect stem, 5 stem, and then simple leaf, or many more characters, then I can easily, by putting this data into a software, I can easily identify, okay, this species belong to this group, or these species belong to that group, okay? So it makes the classification easy. Next one is cytotaxonomy. Cyto means cells, okay? When we're using the cytological information like chromosome number, structure, and behavior for classification of the organism. Chemotaxonomy, when we are using the chemicals which are present in plants, like there are some plants which have flavonoids, there are some plants which have terpenoids, they have alkaloids like morphine, codeines, right? There's a poppy plant, Papaver somniferum, which has opium. So different plants, they have like different chemical, like there are plants which have oxaloacetate, right? Crystals. Next, we are going to discuss algae. Algae, they are chlorophyll-bearing organism because they have chlorophyll, and chlorophyll play a very important role in photosynthesis by trapping the light energy, right? So these algae, they are photosynthetic in nature. They perform photosynthesis, they make their own food, they are autotroph as well. Their body is thalloid in nature. That means it is not differentiated into leaves, root, and stem. They are largely aquatic. It could be fresh water also, or marine water also. They also occur in other habitat like moist stone, soil, and wood. You have, in the raining season, you have observed the algae over the soil, right? So you can observe the algae on the soil and wood as well. Algae, they also shows the symbiotic relationship. If they're showing the symbiotic relationship with fungi, then the association is known as lichen. Okay, and remember the lichen is a very good pollution indicator. Algae, they also shows the symbiotic association with animals. Also, example, the sloth bear. You can see in the image, this is an image of the sloth bear. Here, the bear is appearing green in color because the algae is present over its surface. The algae is getting the shelter, and the bear the bear is providing the shelter to the algae, and the algae in turn imparting the green color to the bear. so that the color of the beer will be green, and it can easily camouflage with the background or the environment, so that it cannot be easily detected or recognized by the predator. Okay, so the slot bear, they can easily escape from the predator by camouflage them. And the algae, they are of various form and they can be of various sizes. They are ranging from colonial, like when the cells they group together, they form a colony, like in volvox. They can be arranged in a series. They can be arranged in a series and they form a filament like Eulothrix and Spirogyra. The algae, they can reproduce by vegetative reproduction also, by showing fragmentation, asexual reproduction also by zoospores, and sexual reproduction also by isogamous and oogamous. We know that in sexual reproduction, the two gamete diffuses, right? And the gametes can be of similar size. If the gametes are of similar size, then the reproduction is isogamous. Iso means same, gamous means gamete. Okay. Remember these two examples. These are very important. All the examples which are given in NCERT are very important. Remember those. Okay. And even the exceptions. They're very, very important from the exam point of view. So here you can see these gametes are similar in size, but they are flagellated. It is found in Eulothrix. Similar in size, but non-flagellated. It's found in Spirogyra. Remember this example. And anisogamous, when the gametes, they are dissimilar in size, it was observed in Eudorina. And the third one is oogamous, when the male gamete, they are small and motile, and the female gamete, they are large and non-motile. It is observed in Volvox and Fucus. Remember these examples, okay? Now let's discuss some of the uses of the algae. Algae, they play a very important role in the ecosystem because they perform photosynthesis, so they can fix at least half of the total carbon dioxide which is present in the earth. So they play a very important role in the ecosystem. Because they are photosynthetic, we know that during the photosynthesis, the oxygen is released. So in the environment where the algae is present, that has an increased level of the dissolved oxygen. Algae, they are also edible also, like Porphyra, Laminaria, Sargassum. They are among the 70 species of marine algae and they can use it as food. Okay. But there are certain algae which produces hydrocolloids. Hydrocolloids are the water holding substances. Like brown algae, they produces algin. Red algae, they produces carrageen. Very, very important. Okay. Remember this point. Very, very important. I'm marking all the important points. Please remember this. Agar is used as a solidifying agent. It is used in the ice cream. It is used in making jellies. It also used in the tissue culture and in laboratory to grow the microbes. Okay. So this agar is obtained from Gelidium and Gracilaria, and they both are the red algae. Remember this, very, very important. Chlorella is a unicellular algae and it is rich in proteins. So it is used as a food supplement by the space travelers. Remember these three examples, very, very important, students. Okay. First, we are going to discuss chlorophyce. That is green algae, and why it is green in color? Because it has the presence of the chlorophyll pigment, which is A and B. They can be unicellular like Chlamydomonas. They can be colonial like Volvox. They can be filamentous like Eulothrix and Spirogyra. These pigments, they are present in the chloroplast, and the shape and the number of the chloroplast also vary. Like it could be 1 in case of Chlamydomonas. It could be 20 to 40 in case of mesophyll cells. So the number of the chloroplast also vary per cell. And the chloroplast shape, it could be discoid, it could be plate-like, it could be reticulate, it could be cup-shaped, okay? They can be of different shapes in different species. These chloroplasts, they have a storage bodies, which are known as pyrenoids, okay? And they contain the protein besides starch. That means they contain proteins as well as the starch. And these pyrenoids, they are present in chloroplast. Remember this. And most member of the green algae, they have these pyrenoids. We know that the reserve food material in green algae is starch, right? But there's some algae which also has oil droplet. The cell wall is made up of inner layer of cellulose and the outer layer is lactose. Vegetative reproduction is by fragmentation, asexual reproduction by zoospores. Sexual reproduction is through isogamous and isogamous or oogamous. And these are the example of green algae. Remember, very, very important point. Remember all the examples, they are very important. Next one is brown algae. Also Phaeophyceae. They're primarily found in marine habitat. They show great variation in size and their forms. They have chlorophyll A, they chlorophyll C, carotenoids, and xanthophyll. Remember this pigments, okay? Important. They have fucoxanthin, because of which they appear brown in color. Remember this. The brown algae, they're appearing brown in color because of the presence of the fucoxanthin. The reserve food material is laminarin and mannitol. In green algae, it was starch. In brown algae, the reserve food material are laminarin and mannitol. Remember this, important. In vegetative cells, they have a cellulosic wall, and the cellulosic wall, they are also covered by a gelatinous coating of algin. Remember this. And very important point that in brown algae, you will observe that the algae, they have the plant body, it is differentiated into holdfast, stipe, and frond. Okay, holdfast is for the attachment to the substratum. Stipe, it is stalk. And frond, they are leaf-like organ which perform photosynthesis. Okay. In fucus, you will observe air bladder. Air bladder is for providing the buoyancy. Vegetative reproduction is by fragmentation. Asexual reproduction is by the zoospores. And these zoospores, very, very important point. This is also a very important point. That in Phaeophyceae, brown algae, the zoospores, they have two flagella. They have unequal flagella. And these flagella, they are lateral in position. Okay. In green algae, they have 2 to 8 flagella. The flagella, they are equal in size. They are apical in position. Remember this flagella number, their position. Okay, it's very important. Sexual reproduction could be isogamous and oogamous. And remember these examples: Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Very, very important. Examples are very important. Okay. Now, next one is the Rhodophyceae. Rhodophyceae, commonly known as red algae. And why this algae is red in color is because of the pigment, which is phycoerythrin. Remember this pigment name. Okay. And the algae, they can be found in both surface. They can be found over the surface of the water, or they can be even found at the depth of the ocean. Okay, where the very little light penetrates. So they can be found in both the regions. Remember, the reserve food material in case of the red algae is the floridine starch. In case of the green algae, it was starch. In case of brown algae, it was laminarin and mannitol. In case of red algae, it is floridine starch. Please remember this reserve food material. It's very important. And this point is also very important that they are similar in structure to the amylopectin and glycogen. Also very important. Remember this. We know that the starch is composed of amylose and amylopectin. Amylose is unbranched. It has alpha-1,4 glycosidic linkage. Amylopectin is branched. It has alpha-1,6 glycosidic linkage. Glycogen is branched. It has alpha-1,4 and alpha-1,6 glycosidic linkage. This I've already taught you in the biomolecules. But here, please remember this reserve food material. Vegetative reproduction is through fragmentation. Remember, in red algae, the flagella are absent. Very, very important point. Here, the flagella are absent. So, the asexual spores, they are also non-motile, and the sexual gametes, they are also non-motile. Remember this. And in green algae and in brown algae, the sexual reproduction is taking place through isogamers, anisogamers, or oogamers, right? But in case of red algae, the sexual reproduction is oogamers. Remember this very, very important point. That here flagella are also absent and the sexual reproduction is oogamers. Remember these examples: Polysiphonia, Porphyra, Gracilaria, and Geladium. And remember, from Gracilaria and Geladium, we obtain agar. You can see this. This is a powder form. This is agar, and it used to make jellies and ice creams, and also used in tissue culture. This table is given in NCERT. It's very, very important because it summarizes most of the important points. Major pigment which are present in algae, like in green algae, the chlorophyll A and B. In brown algae, the chlorophyll A, C, and fucoxanthin. In red algae, there's chlorophyll A, D, and phycoerythrin. Remember the pigment. Storage food. In case of green algae, starch. In case of brown algae, lamitone and mannitol and laminarin. In case of red algae, floridine starch. Here, the cell wall in case of green algae is cellulosic. In brown algae, it is cellulose and algin. Algin is present over the cellulosic wall. Algin is a gelatinous coating. Remember this. In red algae, the cellulose, pectin, and polysulfate ester are present in the cell wall. Please remember the flagella number, their position. It's important. In green algae, it is 2 to 8 in number. They are equal in size. They are apical in position. In brown algae, they are 2 in numbers. They are unequal in size. They are lateral in position. And in red algae, the flagella are absent. Please remember this, that in green algae, the flagella are 2 to 8, equal in size, apical. In brown algae, the flagella are 2, unequal, lateral in position. In red algae, the flagella are absent. It's very, very important point, okay? Please, students, remember these points. Next, we are going to discuss is the life cycle of algae. So in the dominant body of the algae is the gametophyte, and it is haploid in nature. These gametophyte, they are dominant, they are green, they are photosynthetic in nature, right? And these gametophyte, gameto means gamete and the phyte means plant body. So this plant body, it bears the gamete, so it is named as gametophyte. So it have these sex organs are present on the gametophyte, which is antheridium and oogonium. Antheridium is the male sex organ and oogonium is the female sex organ. So antheridium, it produces the anthrozoids, and oogonium, it produces egg. So now anthrozoid and the egg, they get fuses, and the process known as fertilization or syngamy. Zygote is formed, and this zygote is diploid, right? This zygote undergo meiosis and the haploid spores are formed, and this spores on germination again form the gametophyte. You can see the majority of the portion are haploid, right? So it's a haplontic life cycle. Majority of the algae, they show haplontic life cycle, but there are some exceptions are also present, which we are going to discuss later when we are going to study the life cycle pattern or the alteration of generation. Here you can see that there's gametophyte, then there's zygote, and this zygote, the single cell zygote represent the sporophyte in case of algae. Okay. And one more important point here, these sex organs, they are unicellular and non-jacketed. Non-jacketed means that there is no protective covering is present around these sex organs. So they're unicellular and they're non-jacketed. Remember this. Next, we are going to discuss bryophyte. Student, I'm going fast because this chapter is very vast, okay? Next, Now we are going to discuss bryophytes. Bryophytes are found in moist, shaded areas. These are known as amphibians of the plant kingdom. Why amphibians? Because they can grow in soil, but they require water for their fertilization. So they are known as amphibians of the plant kingdom. They also play a very important role in plant succession. Remember, during when we discussed the Zirac succession, the succession which occurs on the dry areas, when we study the succession on the bare rocks. So there's a pioneer species which are lichen, and these pioneer species, they are also followed by the mosses. So the mosses, they also play a very important role in the plant succession on bare rocks. They do not have the true roots, stems, or leaves. They have a root-like structure, which are rhizoids. They have leaf-like structure, they have stem-like structure, but they does not have the true root, stems, or leaf. Remember this. Their plant body is gametophyte, and it is haploid. And this gametophyte is a dominant plant body. It is independent. That means it is free living, and it is photosynthetic in nature. This gametophyte, it bears the sex organ, and those sex organs, they are multicellular in nature. In algae, the sex organs were unicellular. Here they are multicellular, and they are jacketed as well. The male sex organs are antheridium, and these antheridium, they produces the biflagellated antherozoids. Female sex organs, they are archegonium, and archegonium is flask shaped. It produces a single egg. So these antherozoids, they are released into the water when they come in contact with the archegonium, which has the egg. These antherozoids, when come in contact with the egg, they get fuses with the egg, and they produce a zygote. And this zygote, it produces a sporophyte. Okay. In algae, the zygote was undergoing the meiosis and produces spores, and the spores are forming the gametophyte, right? But in bryophyte, the zygote, they will produce a multicellular sporophyte, and this sporophyte is differentiated into foot, seta, and capsule. Okay, and this sporophyte is forming on the gametophyte. Okay, so it is dependent on the gametophyte for its nourishment and development. We know that the sporophyte is differentiated into foot, seta, and capsule, right? Foot is for the attachment, seta is the stalk, and the capsule, it bears the spore mother cells. The meiosis occurs in the spore mother cells, and the haploid spores are formed, which on germination produces the gametophyte. Okay? You can see in the image, this is a sporophyte, this is a gametophyte. And the bryophyte, it includes liverworts and mosses. There's some mosses which can be used as a food for the herbaceous mammals, birds, and other animals. The sphagnum are present. Sphagnum is also a moss. And when the sphagnum, they are partially decomposed, they form a peat. Peat is used as a fuel. Very important, okay? Many times this question asks that peat is obtained from, remember, is sphagnum. And we know that the mosses, they have a high water holding capacity. So they are used as a packaging material for transshipment of the living materials. Okay. And the mosses, they form a dense mat on the soil. Okay. So the rain, when the rain falls, it does not affect the soil directly, and the soil erosion does not take place. So mosses, they help in the prevention of the soil erosion. Bryophytes, it includes liverworts and mosses. First, we are going to discuss liverworts. They grow in moist, shady habitats such as bank of streams, marshy ground, deep in the woods. In liverwort, the plant body is thalloid, which example is Markensia. You can see this Markensia. Their body is dorsiventral. Dorsal is the upper surface, ventral is the lower surface. Okay. Its dorsiventral nature is closely appressed to the substratum. Appressed means they are flattened. Okay, the flattened. Asexual reproduction is by gamma. You can see this. These are the gamma cup. Asexual reproduction occurs through these gamma cup. They are green in color. They are multicellular. They are the asexual birds. These gamma, they get detached from the main plant body, and they can grow and form into a new individual. So these gamma cup, they help in the asexual reproduction. Remember this is important, okay? And Markensia, they are unisexual in nature. They are dioecious. You can see this. There's a female thallus and the male thallus separately. This is a female thallus because it's having the archegoniophore. Archegoniophore, this is the stalk which bears the archegonia. Here you can see the next is one is the male thallus, which has enthridiophore. Enthridiophore is the stalk which bears the enthridium, the male sex organ. So mostly the bryophytes, they are bisexual, but the exception is Markensia. Markensia is unisexual or dioecious. And the sporophyte, we know that it is differentiated into foot, seta, and capsule. The capsule, it has the spore mother cell which undergo meiosis and produces the haploid spores. And these haploid spores on germination, they produce a free living, the independent gametophyte. Next one is mosses. In mosses, the gametophyte consists of two stages. Okay. Wait, let me show you the life cycle first. Then it will become more clear. We know in bryophyte, the main plant body is the gametophyte, which is dominant, right? And this gametophyte is haploid. This gametophyte, it bears the sex organs, which are multicellular. The male sex organ is enthridium. The female sex organ is archegonium. Enthridium, it produces the anthrozoid. Archegonium produces the egg. Okay. When the anthrozoid, the male gamete and the female gamete fuses, they produces a zygote, which is 2n in number. This zygote undergo differentiation and produces a sporophyte. And this sporophyte, this zygote, it produces a multicellular sporophyte, and this sporophyte undergo differentiation and produces the foot, seta, and capsule. Okay. The sporophyte, it undergoes the differentiation and it produces the foot, seta, and capsule. Foot is for the attachment, seta is the for the stalk, and there's a capsule. Let me draw it. Let's say it's a foot, this is a seta, and this is a capsule. Like this. And inside the capsule, there are spore mother cells are present. And these spore mother cells, because they are diploid, they undergo meiosis, and the haploid spores are produced. Okay, these haploid spores, in case of the liverwort, they are directly germinated into the gametophyte. They undergo germination and they are directly forming the gametophyte. But in case of mosses, these haploid spores, they first form the primary protonema, like this. This is a spore. It is germinating. First, it will form a protonema. Primary protonema. Let's say it's a primary protonema. Now from the primary protonema, now this is branch, let's say it's secondary protonema. And this secondary protonema, it bears the lateral buds. And from this lateral bud, the leafy gametophyte arises. Like this. This is the leafy gametophyte. So in case of mosses, suppose they are first forming the primary protonema, then forming the secondary protonema, and secondary protonema, they have lateral buds. And these lateral buds on germination, they produce a leafy gametophyte. And remember, the leafy gametophyte, it bears the sex organ. That means this. I hope it is clear. This is what the same thing is written in this slide as well, that the gametophyte, it consists of two stages. The first stage is the protonema stage, which develops from the spore. It is green, it is branched, it is frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as lateral buds. Okay. And this leafy gametophyte, which bears the sex organs. This I've already told you. Please remember the example, that Pleureria, Poliotrichum, sphagnum. These examples are very important. Please remember all the examples. This is the image of Punaria. You can see in the image, the rhizoids are present. This is the main axis. And you can see this leaf. This is the leafy gametophyte. And this leafy gametophyte, they bear the sex organs. Okay. And when the bear the sex organ like antheridium and archegonium, and when the male and female gamete diffuses, they form a zygote, and the zygote undergo they form the sporophyte, and the sporophyte is differentiated into foot and the seta, and there is a capsule. This is a sporophyte, and you can see the sporophyte is forming on the gametophyte. That means the sporophyte is dependent on the gametophyte. Okay, remember this, it's very important. Okay, now you can see. This is a sporophyte, and this sporophyte is dependent on the gametophyte for its nourishment and development. Okay, remember this point. It's an important point in. It's a very important point in bryophyte, that the sporophyte is dependent on the gametophyte. Okay, it's very important point. Please remember this, student. And here the gametophyte is free living. That means it is independent, it is dominant, it is green in color, it is photosynthetic in nature. But the sporophyte, it is dependent on the gametophyte. Okay, remember that the sporophyte is not free living, it is dependent on the gametophyte. This I've already discussed. Next one is the pteridophyte. We know that in bryophyte, the vascular tissues are absent, but in pteridophyte, the vascular tissues are found and they're developed. Here, the plant body, it is differentiated into the true root, stems, and leaf. Remember this. In bryophyte, the plant body is not differentiated into true root, stems, and leaf. But in pteridophyte, the plant body is differentiated into true root, stems, and leaves. Let me show you the life cycle and become easy. In pteridophyte, the main plant body is the sporophyte. Okay, remember this. The main plant body in case of pteridophyte is sporophyte. In bryophyte, the main plant body is gametophyte. In algae, the main plant body is gametophyte. But in pteridophyte, the main plant body is the sporophyte. And these sporophyte, they have photosynthetic leaves also, which perform photosynthesis, and they have reproductive leaves also, which are sporophylls. And these sporophylls, they have sporangia. Okay, they have sporangia. And these sporangia, they have spore mother cells. So when meiosis occurs in spore mother cell, the spores are formed, haploid spores are formed. And these haploid spores, on germination, they produce a gametophyte. And this gametophyte, it is small in size, it is green, and it is independent. Okay, and this gametophyte is known as prothallus. Remember this, in pteridophyte, remember prothallus. In mosses or bryophyte, please remember protonema. It's important that protonema you have seen in or observe in case of mosses, but prothallus in case of pteridophyta. So these spores on germination, they produce a gametophyte, and the gametophyte is known as prothallus. This gametophyte, it bears these sex organs, which are antheridium and archegonium. Antheridium, they produce anthrozoid. Archegonium, they have egg. And this male and female gamete, after fusion, they produce a zygote. This zygote undergo mitosis and form embryo, then embryo again form the sporophyte. This is the whole life cycle. Okay. And in bryophyta and pteridophyta, you have observed both sporophyte and the gametophytic phases, right? And they're having an equal equal duration, on almost an equal duration. So we call that the bryophyte and the pteridophyte, they have a haplodiplontic life cycle. Okay. Algae, most of the algae, they have haplontic life cycle, but bryophytes and pteridophytes, they have haplodiplontic life cycle. Remember this, very, very important. Now let's discuss some other important points. Nourishing, see, here there's a sporophyte which is diploid. There's sporophylls, there's sporangia. These are also diploid. Four mother cells, they are diploid. But the spores, you can see spores, they are haploid. So this phase, this is the haploid phase, right? This zygote is diploid. So you can see this is the diploid phase, this is the haploid phase. So that is why the life cycle is known as haplodiplontic. But in case of algae, the zygote, which is a single cell zygote, is representing the sporophyte. See, let me show you the algae. Here you can see the diploid phase is this much small. The single cell zygote is representing the sporophyte, which is very small. Okay, and the major portion is the the haploid. So this is why this life cycle is haplontic. Here you will observe this is diploid, this is haploid. So here the life cycle is haplodiplontic. Please remember one more important point. This sometimes it is asked that the majority of the pteridophyte, they are homosporous. That means they produce the same type of spores or the similar spores. But Selaginella and Salvinia, they are heterosporous in nature. Remember this. And the pteridophyte, they are the precursor of the seed habit. The seed is not formed in pteridophyte, remember, but they are the precursor of the seed habit, because the development of the zygote into the young embryo takes place within the female gametophyte. That means this gametophyte, it remains attached to the plane, remain attached to the parent. Okay, so it retain on the parent sporophyte for variable period. So it's a precursor for the seed habit. Remember, this is important. Okay. Next one is gymnosperm. Gymno means naked and sperms means seed. So there's gymnosperm, the naked seeds are formed. That means the ovule is not enclosed by the ovary. Okay, remember this. Gymnosperm, they are usually consist of medium-sized trees, tall trees and shrubs. They never include herbs. Remember this, the gymnosperm, it includes medium-sized trees, tall trees and shrubs, but never herbs. And you can see in the image, these are the sequoia tree, which are the tallest and very giant tree. Remember this. You can see in the image how tall they are. And the roots are generally taproot. In gymnosperm, we are going to study Pinus and Cycas. The roots of the Pinus, they show symbiotic association with the fungi, and the association is known as mycorrhiza. Remember this, the Pinus, it shows an association with the fungi, and the association is known as mycorrhiza. In Cycas, you will observe that the colloid roots of Cycas, they show a symbiotic association with the cyanobacteria, which are a nitrogen fixers like Nostoc and Anabaena. Remember this, it's also important. And the stems are unbranched in Cycas, and the stems are branched in Pinus and Cedrus. This is also important points. And one more important point, that the leaves of the gymnosperms, they are well adapted to the environment. So they're well adapted to withstand or overcome or tolerate the extreme temperature, humidity, and wind. Their leaves, they are needle-like, so they can reduce the surface, so the less loss of water will take place. They have thick cuticle, That means they're and cuticle is a waxy layer. So that means they're preventing the loss of water. They have sunken stomata. We know that maximum transpiration takes place through the stomata, right? So sunken stomata, that means that they also help in reducing the water loss. So needle-like leaves, thick cuticle, sunken stomata, they all help in reducing the water loss. Okay, please remember this point that the leaves in gymnosperms, they are well adapted to withstand the extreme of temperature, humidity, and wind. Gymnosperm, they are heterosporous. Remember this, they have microspores and megaspores. Let me show you the life cycle, then it becomes very easy. Here also the main plant body is diploid or sporophyte. The sporophyte, they have microsporophyll and they have megasporophyll. We know that these sporophylls, they cluster together and they form a cone like this. They all the microsporophyll, like if they cluster together, they will form the male cone. Okay, and in some the megasporophyll, they will cluster together and they will form the female cone. The male cone, they have microsporangia. Female cone, they will have megasporangia. The microsporangia, they will have microspore mother cell, which undergo meiosis and they will produce haploid microspores. And these haploid microspores on maturation will produce the pollen grain, which is a male gametophyte. In here, the megasporophyll are present, which cluster together and form the female cone. They have megasporangia. Megasporangium will have the megasporangium is also referred as a ovule. Okay, but in case of gymnosperm, the ovule is unitegment. They have only a single integument. Remember this. And the one cell of the nucellus, it gets differentiated into the megaspore mother cell. This megaspore mother cell, it undergo meiosis and produce the 4 haploid megaspores. Out of 4, 3 will get degenerate and only 1 is functional. Okay. Now this functional megaspore will form the female gametophyte and the female gametophyte will bear the archegonium. Okay. And this pollen grain, they will produce the male gamete. When the pollen grain, the pollination case of gymnosperm, they are usually the wind pollination. Remember this. And the pollen grains, when they reach the reach the archegonium, the male gamete are released, and these gamete diffuses with the egg. They form a zygote, and these zygote then undergo mitosis division. They form embryo, and the embryo. Now the ovule get mature, and now the ovule will form the seed, but the seed is naked because the ovule is not enclosed by the ovary. Okay, remember this. And one more important point, the cycas is unisexual or dioecious, but Pinus is bisexual or monoecious. I've almost covered all the points with the help of life cycle. Next one is angiosperm. We have already discussed the angiosperm in detail during the sexual reproduction in the flowering plants, right? But I'll give you a quick review. This is the main plant body in angiosperm is also the sporophyte. They have the flower. Angiosperm, they are the flower bearing. Remember this, the flower, in the flower, there's the androecium is present, gynoecium is present. Like androecium, they have stamens, and the stamens, they have anther. In the anther, microsporangiae are present, which have a microspore mother cells. These microspore mother cells, they undergo meiosis and they produce the microspores. And these microspores on maturation, they will produce the pollen grains. And the pollen grains, they have 2 layers, the exine and intine, right? And there's a germ pore is present through which the pollen tube are forming. And here, there's a female reproductive organ, there's a stigma is there, the style is there, and the ovary is there. And the ovule are present. Inside the ovule, nucellus are present. When the one cell of the nucellus, they get differentiated into megaspore mother cells. These megaspore mother cells, they undergo meiosis and produce the haploid megaspore. Out of four, three get degenerated and only one is functional. That functional megaspore form the female gametophyte, and here the female gametophyte is known as embryo sac. It is 8 nucleated and 7 celled. It has the egg apparatus, which consists of 2 synergid and 1 egg. There's a central cell is present, which consists of 2 polar nuclei, and there's a 3 antipodal cells are present. Now here you can see this pollen tube. Here pollen tube is formed and it having the 2 male gametes. This pollen tube will enter into through the micropylar end. Okay, will enter through the micropylar end. These male gamete will enter into the embryo sac, and the direction or the direction of the pollen tube, it is guided by the filiform apparatus, which are present in the synergid. One gamete will fuse with the egg, they will form a zygote. The other male gamete will fuses with the central polar nuclei, and they will form the endosperm, which is a primary endosperm. Okay. Now this zygote will undergo mitosis division. They will form the embryo. And this embryo, again, they will form the sporophyte. Next is the life cycle pattern. Most of the algae, I told you that they have the haplontic life cycles. Okay. The sporophytic generation is represented by the one cell zygote. The sporophytic generation is for a very short duration. Okay. And the gametophytic, or which is the dominant and photosynthetic phase, is occupied by the gametophyte. The example are volvox, Spirogyra, and Chlamydomonas. They are the example of the haplontic life cycle. Remember that most of the algae, they show haplontic life cycle, but there are some algae like fucus which show diplontic life cycle. There's algae like ectocarpus, Polysiphonia, kelps. They show haplodiplontic life cycle. That is why we say that most of the algae, they show haplontic life cycle. We don't say that all the algae, they show haplontic life cycle, because there are some exceptions. Exception is fucus, they show diplontic life cycle. Exception is ectocarpus, Polysiphonia, and kelps, which are haplodiplontic. They show haplodiplontic life cycle. Please remember these exceptions. These are very important. Okay. And the diplontic life cycle you have also observed in case of gymnosperm and angiosperm. Here the gametophytic phase is represented by the fuse-celled haploid gametophyte. Okay. And the major portion is occupied by the sporophyte, which is diploid. So it's a diplontic life cycle. Haplodiplontic you have observed in case of bryophytes and pteridophyta. In bryophyte, the gametophyte is dominant, independent, photosynthetic, and sporophyte is dependent on the gametophyte. Remember this. In pteridophyta, sporophyte is dominant, independent, photosynthetic, vascular plant body, but the gametophyte is independent, but it is short-lived. Remember, in pteridophyta, the gametophyte is prothallus. Remember all the example. Every example is very important. Now, let's take some question. Question number 1 says the life cycle of ectocarpus and fucus respectively are. Option 1 is diplontic or haplodiplontic. Option 2 is haplodiplontic, diplontic. Option 3 is haplodiplontic, haplontic. Option 4 is haplontic and diplontic. Just now I've told you. Very good, Shishant. Very good, Veena. Very good, Shishni. All of you have answered it correctly. Life cycle of Ectocarpus. Ectocarpus brown algae, it is haplodiplontic. Fucus, it is diplontic. It's also brown algae. So the correct option is 2. Gamma are present in 1 mosses, 2 pteridophytes, 3 some gymnosperm, 4 some liverworts. Remember, gamma, they are involved in asexual reproduction. They are the asexual birds. Very good, Veena. Very good, Shishmi. Very good, Ritu. You will observe the gamma in Markensia. When Markensia is a liverwort. Please remember, it's not a moss, it's a liverwort. And the gamma, they are involved in asexual reproduction. For example, if this is a thalli, like this, these are the gamma curves. They are the rhizoids. When they get detached, they can form a new individual. So the correct answer is 4th, that is some liverworts. Question number 3 says, which of the following statement is correct? 1 is ovules are not enclosed by ovary wall in gymnosperms. Option 2 is Selaginella is heterosporous while Salvinia is homosporous. Option 3 is host tails are gymnosperms. Option 4 is stems are usually unbranched in both Cycas and Cedrus. What do you think? What will be the correct option? They're asking which of the following statement is correct. Very good, Shishni. Very good, Veena. Question is asking which of the following statement is correct. Okay. Ovules are not enclosed by ovary wall in gymnosperm. Yes, that is why they have a naked seed. Because the ovules, they are not enclosed by the ovary wall. Selaginella is heterosporous. Yes. Salvinia is homosporous. No. Salvinia and Selaginella, the both are heterosporous. Remember this. Host tail are gymnosperm. No. Host tail are pteridophytes. That's why I told you examples are very important. Please remember all the examples. Stems are usually unbranched in both Cycas and Cedrus. Stem is unbranched in Cycas, but in Cedrus and Pinus, the stem is branched. Okay, so the correct option is option number 1. I hope, Sushant, it is clear now. It's already 1. We are winding up the class. Thank you all for your participation.